Alright, ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here. ETM Hotel, Rna, Shawn, welcome in peace. My name is Shawn. I'd like to thank everybody tuning into Science with Shawn. Brought to you, sponsored by Spears of the Mossi, Volume 1, Spiritual Combat, Volume 3, and From Spears to Pen, Volume 2. Yep. This is brought to you by the Mossi Warrior Clan and Kofi Pi-Side Research Team. How is everybody doing this evening? Like the show, share the show. Like the show, share the show. We're about to get it in. Not going to hold y'all too long. We're going to come back. Now, we talked about this the other day about the new strand. Everybody making a big deal out of it. So, let's get to it. It's late. I only spent a lot of people on here. But let's start off with an article. And then we're going to deal with what we need to deal with. Let's see what NPR talking about right now.
Because these are hard.
Okay, they ain't talking about nothing right now. Let's see if we can get a couple more people in here. Bear with me, I'm sharing the show. What are y'all talking about? Man, I ain't even gonna get to deal with NPR. I'm gonna get straight to the straight. Straight to the straight. We're gonna come back to this article right here. But let's talk to my man real quick. Yeah, let's get in with Vince. Let's see what Vince talking about, ladies and gentlemen. He about 20 minutes, but let's deal with it.
I think the mutating. What does that mean for us? Well, actually, it probably doesn't mean very much. I would say this is not a headline because viral genomes are always mutating. Yep. I think the headline writer needs to bone up on his or her virology. So let's do that first before we talk about this variant. By the way, this headline is in reference to the variant in the UK. What is mutation? Let's define it because most people don't seem to get it right. This is a definition of mutation from Principles of Virology, the 5th edition, a virology textbook of which I am a co-author. And here we go. A mutation is a change in DNA or RNA comprising base changes and nucleotide additions, deletions, and rearrangements. And these changes, when these mutations occur in open reading frames, they can be manifested as changes in the proteins. So one or more base changes can produce a single amino acid substitution, a truncated protein, or no protein. The terms mutation and deletion are used incorrectly or ambiguously to describe alterations in proteins. That is what the headline writer did. They used mutation to describe changes in protein. Viruses mutate all the time. Whether those mutations mean anything is the real question. Let's take a little bit closer look at that first. We know that genome mutation rates vary. Every genome, whether it's a human genome or a fly genome or a virus genome, a DNA virus and RNA virus, they all undergo mutations at every reproduction cycle because the enzymes that carry out replication of genomes make mistakes. Some organisms can correct the mistakes, but others can't. And this is a very informative graph. We're looking at genome mutation rate of different kinds of nucleic acids. And the mutation rate here is on the y-axis. It's substitutions per nucleotide per generation. In other words, every time the nucleic acid is copied, how many changes do we see per nucleotide? Coronaviruses, of which SARS-CoV-2 is a member, of course, single-stranded positive sense RNA viruses. So those are shown here in yellow circles. And in general, these plus-stranded RNA viruses, their mutations rate can vary from 10 to the minus 3. That is 1 in 1,000 changes per nucleotide per generation, all the way down to about between 10 to the minus 5 and 10 to the minus 6, 1 in 100,000 or 1 in a million, much less. And there are others in between as well. And you can see that bacteria have very low mutation rates. They have DNA genomes. The DNA enzymes can correct mutations. And viroids have very high mutation rates. So the RNA viruses we're talking about today are right in the middle here, ranging in mutation rates anywhere from 1 in 1,000 to about 1 in a million. Now, most RNA viruses don't have any ways to correct the mutations that the polymerases make. These are viral enzymes, of course. The coronaviruses are different. They have very large RNA genomes, 30,000 bases, and they have an error correction machinery. So their mutation rate is slightly less. However, it is still probably around 1 mutation per 10,000 or 20,000 bases per genome replication. You know, in an infected cell, if you make 10,000 genomes, let's say 10,000 base RNA genome where the frequency is 1 in 10,000 per reproduction cycle, you're going to make easily 10,000 RNAs in an infected cell. So basically every base in the viral genome can be substituted. That's mutation. It happens all the time. And so
hold on. Vince, you going in. All we hearing is RNA. We hearing you talk about the cells reproducing RNA, and then guess what? We got an mRNA vaccine to help train the cells to create a defense mechanism against SARS-CoV-2, the S protein. Did you know that your body continues to make the RNA? So it's only right to deal with the mRNA vaccine so that you can fight off the spike from SARS-CoV-2? Go back in, Vince. They ain't hearing us, Vince. They ain't hearing me, you, none of that. Let's get back to your chart. Give it to us again, Vince.
And the mutation rate here is on the y-axis. It's substitutions per nucleotide per generation. In other words, every time the nucleic acid is copied, how many changes do we see per nucleotide? Coronaviruses, of which SARS-CoV-2 is a member, of course, single-stranded positive sense RNA viruses. So those are shown here in yellow circles. And in general, these plus-stranded RNA viruses, their mutations rate can vary from 10 to the minus 3. That is 1 in 1,000 changes per nucleotide per generation, all the way down to about between 10 to the -5 and 10 to the -6, 1 in 100,000 or 1 in a million, much less. And there are others in between as well. And you can see that bacteria have very low mutation rates. They have DNA genomes. The DNA enzymes can correct mutations. And viroids have very high mutation rates. So the RNA viruses we're talking about today are right in the middle here, ranging in mutation rates anywhere from 1 in 1,000 to about 1 in a million. Now, most RNA viruses don't have any ways to correct the mutations that the polymerases make. These are viral enzymes, of course. The coronaviruses are different. They have very large RNA genomes, 30,000 bases, and they have an error correction machinery. So their mutation rate is slightly less. However, it is still probably around 1 mutation per 10,000 or 20,000 bases per genome replication. In an infected cell, if you make 10,000 genomes, let's say 10,000 base RNA genome where the frequency is 1 in 10,000 per reproduction cycle, you're going to make easily 10,000 RNAs in an infected cell. So basically every base in the viral genome can be substituted. That's mutation. It happens all the time. And so let's look at it in terms of SARS-CoV-2 or coronaviruses. These viruses get into cells, their RNA is translated, and then among the proteins made are those that are needed to do genome replication. You go from a plus strand to a minus strand to more plus strands. And of course, those plus strands eventually end up in new virus particles. Every time the genome replicates from plus to minus, from minus to plus, there's a certain frequency of errors being made. So all the viruses come out, and there are many thousands of viruses per cell that are made, constitute a collection of mutants, which we call a quasi-species. For SARS-CoV-2, again, with a RNA genome of 30,000 bases, all the viruses that come out of a single cell in your upper respiratory tract, there are many, many different mutants. It's a collection of mutants schematized by this diagram on the right, where every line is a virus genome and the symbols are different mutations. Every genome can be different. Not all of those viruses with mutant genomes make it. Some of the mutations are lethal. They destroy the ability of the virus to reproduce the next time it infects a cell. Many of them are neutral. They do nothing. And some of them may have an impact. So far with SARS-CoV-2, the impacts have been minimal. I haven't seen any that really have changed the course of disease. If we had one that changed the property, it would be called a strain, but we have no new strains yet. In my opinion, only variants. SARS-CoV-2 genome is always mutating. Doesn't matter. The real question is whether the mutations are causing amino acid changes in proteins that make a difference. Just to give you a sense of how many mutations we've seen, this slide was made, about a month or two ago, where in over 90,000 isolates, we have seen 12,000 mutations. And any two of those isolates differ by about 10 bases. So 10 out of 30,000 bases differ. And many of these have no consequence. They're actually just markers that we can use to do contact tracing. They have no effect on the way the virus reproduces. None of these mutations have led to a new strain. A strain is a variant that has a distinct biological property. And I mean in people. You can look in cells in the laboratory and find all kinds of differences, but what matters is in people. And as far as I'm concerned, none of the isolates so far have proven implications for human transmission or pathogenesis, including the latest variant isolated from the UK, which we'll get into detail in just a moment. It's very difficult to use epidemiological data, that is, looking at the spread of a virus in a population and conclude that a particular variant is spreading better. And here's why. It's because of something called the founder effect. So here on the left is our original population of viruses reproducing in an individual, say. And remember, because of all the mutations that go on, the viruses that come out of that person are a mixture of all different viruses, like we showed in this slide here. Every genome is different. But when that person transmits to another, it may not transmit all of them. Maybe a subset will be transmitted, shown here by the green and orange viruses, which have a particular set of mutations. And so now that virus is in the population, and everyone infected by it will contain those changes as well. So that's cool.
All right. Let's see if I can dumb this down. I'm going to try my best to dumb this down. He is basically saying that he's not worried about the new variant because it's not a strand. If it is a strand, then it is an issue because a strand has more of effect on us than a variant. Okay? Ladies and gentlemen, he is saying that this new variant, let me see if I can make this make sense. All right. Everybody know what a Chevy Impala look like, right? And like every 4 years or so, Chevy changes the body style somehow, right? You go from, and I'm gonna go back just a little bit. Everybody know what a box Chevy look like, right? From there, you get, you know, the Chevy Caprice. All right, yeah, all right. Okay, I said Caprice. Okay. Then you get the box Chevy Caprice evolved into like a bubble Caprice. Y'all remember that? Because out of that, the new SS was a variant of the Caprice. I'm talking cars. I'm gonna change it up a little bit and maybe it'll make sense. So basically what he's saying is the same thing. It is only different if it changes. If it's still a box Chevy, it's still a box Chevy. It is no longer a box Chevy when it evolved into an oval or into what it looked like today. That will be a new strain. Okay, ladies, let's deal with it this way. What kind of weave do you wear or wig? Is it Indian? Is it Brazilian? Right? Now, I ain't too up on the weave, but y'all know what I'm getting at. There's a strand of weave that costs a lot of money. Then there's another one that don't cost so much. But if you got two different variants of the same weave, if it came from India, like, you might have like a dark color, and then you might have like a lighter brownish brunette color. From both samples come from India. But if it's that Brazilian, it ain't like that India. No matter the length, no matter how it look, they can look a little similar or whatever. That Brazilian might just be a little bit better. That's all he's saying. I try to make it dumb. It's changing. When it changes and evolves into something else, it's a strand. If it's just a variant, it ain't gonna phase us whatsoever. He went into your RNA in your skin, talking about your, the RNA replicating itself. Dead cell. He's basically telling you that cells die right now. You don't contract no disease or whatever. You still got cells that's in your body that's gonna die, and the RNA helps it replicate itself, new cell membrane, so on and so forth. Long story. Real geeky stuff. But this variant is not. Yeah, it might help transmit faster because it's a new variant, but it's not a new strand. If it's a new strand, if it mutates into a new strand, it could be deadly. Don't be laughing at my analogies, man. That's it. It's the same virus. Same. Chavis just said it. He said it time and time and time over. That's it. It just got a him in now. Yep. That's it. It's the Caprice bubble, and the SS look just alike, except for the SS got a little kit on it, and a V8. The Caprice got a V6. That's it. Same thing with the if you want to use whatever your favorite car is. If you got your favorite SS Impala, and then you got, well, now it's LT and then LTZ, or they got LT 1, 2, and 3 and LTZ. You know what I'm saying? So that's it. It looks the same, just an upgrade. The upgrade is a faster engine. That's it. All right. So that's why he's not fearful of it. When it replicates into a new strand or mutates into a new strand, we need to know what the hell going on.
Called the founder effect. And the changes just go along. Coincidentally, they're neutral, they have no effect, and they seem to spread rapidly to a lot of people. That alarms people, but in fact, it's meaningless. And one of the ways you can look at this is because we know that SARS-CoV-2 spreading, when it happens, is actually done by very few individuals. The rapid spread of a virus doesn't have to be a consequence of novel genome mutations. We know that transmission of SARS-CoV-2 occurs mainly by super spreader events. 80% of transmissions are caused by 10% of infectious individuals. This has been studied in a number of countries. Here's a paper out of Hong Kong where they identified the main transmission events were super spreader events in weddings and churches and bars and restaurants, for example. And so here on the very left in A is an example of a super spreader event. You have one individual, B, that went to a bar or a place with a lot of people, infected many others, who then go on and infect many others, and so on and so forth. So you can see, I think, from this, that if individual B in this diagram just happened to have a particular mutation in its genome that, say, led to a spike amino acid change, he or she would spread it to a lot of other people who would spread it to a lot of other people. And eventually it would be in a lot of people, and that's why we get scared when these kinds of events happen. But in fact, those changes can be neutral. They may not be neutral, but you better have some good data to distinguish between them being neutral and not. And in my opinion, we do not have such data yet. So I think anytime a particular amino acid change in the spike, which is what we're seeing now for the UK variant, whenever they spread in the population, it's simply a neutral spread. It's by this founder effect combined with super spreader events. All right, so what about the UK variant? What's going on there? Let's look at the data. The first is a place where you should go to look at new sequence data. It's called GISAID, the Global Initiative on Sharing All Influenza Data. It's basically a website, gisaid.org, where you can go to. It was originally established to share influenza data, but now, of course, SARS-CoV-2 data are being shared. And whenever a new genome or genomes are sequenced, they're deposited here. So here's the new variant. That's the name of it. And the name comes from variant under investigation year 2020, month 12, variant number 1. And this variant was defined by multiple spike changes. They're not mutations. These are the wording from the GSAID. Even them get it wrong. These are spike protein amino acid changes. The mutation is in the genome. And you may say, oh, Vinny, you're being pedantic. You have to get your terms right. Otherwise, people like headline writers for newspapers get confused. Here are the spike protein amino acid changes. There are 2 different deletions, and then 1, 2, 3, 4, 5, 6, 7 single amino acid changes. So that's all the changes in the spike protein of this variant. And they go on further to say an increasing fraction in southern England share several of these, and a handful have been seen through imports in other countries. And what I'm going to do now is show you where these are located. So remember, the viral particle on the lower left is studied with spike glycoproteins that attach to the cell receptor. They're very important. We're making antibodies against the spike in our vaccines to prevent infection. So the spike is biologically very important for the virus. And that spike is a trimer of 3 polypeptide chains. And the structure of that is shown on the right 2 panels here. So we're looking at down from the top at the spike trimer. So you can see from the schematic on the lower left, it's a trimer. And if you look down from the top, you have 1, 2, 3 polypeptide chains. And on the right, we're looking from the side. And here in green is ACE2, the cell receptor. So in the light gray is the part of the spike that binds ACE2, you can see here. Okay, so we're going to look at these changes. The changes in the spike in this variant are in different colors, as you can see down here, cyan, orange or gold, and blue. Let's take a look at where some of these changes are in the spike and what they might mean for its function. So the blue are these single amino acid changes down here. There is one N501Y, which is in gold right there. And then in cyan, there are the deletions. And if you look at the right-hand panel here where ACE2 is binding to spike, you can see that one of these deletions is in the area of where the spike binds. So it could change binding in some way. Based on the evaluation of effect on virus structure and function, the most relevant of all these changes might be N501Y, disparaging to tyrosine at amino acid 501 of the spike. That's in orange here. And here, there's three of them. So 1, 2, 3, and you can see this is potentially impacting the spike. And then they say in positions contributing to potential spike surface variation, and there's a deletion at Y145. One of these cyan deletions is where some antibodies, like one called 4A8, bind. All right, so the others they think have no apparent interest. So the ones they're interested in is N501Y, that could affect the binding of host receptors and antibodies, and then the deletion where one of them in particular could affect an antibody binding site. So here they say the other mutations, the blue ones. So we've focused on cyan and gold. So the blue mutations, they say their effect on structure and function is less clear. And then they also say in another protein called ORF8, there's a early stop codon that could be relevant. This causes a deletion in the ORF8 protein, and these have been seen before in other viruses isolated from Singapore and also other countries. They may actually have a role to play in attenuation. They may make the virus less virulent, which is not something you're hearing at all. All you're hearing is increased transmission. And finally, they conclude, as seen on many occasions before, mutations are naturally expected for viruses and are most often simply neutral regional markers useful for contact tracing. As I've just told you, mutations are naturally expected for viruses. The mutations seen have rarely been affecting viral fitness and almost never affect clinical outcome, but the detailed effects of these mutations remain to be determined fully. And that's a reasonable statement. In the past, we haven't seen any such changes make a big difference in transmission and disease in humans. But we should be aware of them going forward and make sure we study them. And I think that's perfectly fine. But what is not fine is all the hype now, which is about this strain having higher transmission, which simply can't be concluded so far. There's one other document I want to refer to. And again, the URL for it is down here. It's from a committee called the New and Emerging Respiratory Virus Threats Advisory Group. So they had a meeting on the 20th of December. And here are the individuals in this meeting, some of whom are virologists, epidemiologists, and so forth. And they have looked at the data on this variant, and they've made some conclusions. So let me go through them with you, because I think this document is what's scaring the world at the moment, and I don't think it is justified. So I'm going to label these nerve tag considerations. First, within the UK, the variant is mostly in London, southeast and east of England, but has been detected in various parts, but it's mostly in those other areas. And a few have been reported internationally. One confirmed export from the UK to Australia. I would say it's already out because, as they say here, a lot of other countries have lower sequencing capability than the UK and other countries. So it's not easy to know that the variant is already there because you have to sequence isolates from people to know this. Now, here are the data which lead the committee to be somewhat worried. First of all, study is...
Hold on real quick. Let me go on my notes. I need to get this. I need to get this for y'all. Nerve tag right here. This is what we're dealing with. It's a large document. I don't know if I'll be able to fit all of this into the chat, but I'm gonna try. But it will be in the description. It did go in the chat. It may come up as 2 messages. It will be in the description because I will add it in the GSAT. So we will have everything that we need to be knowledgeable on our build. So let's get back.
Correlation between R values and detection of the variant, which suggests an absolute increase in the R value of between 0.39 to 0.93. So R, of course, is the reproductive index, the likelihood that an infected person on average will infect so many other people, right? So for SARS-CoV-2, we think it's between 2 and 3 without mitigation, without doing any lockdown or any kind of masking or distancing and so forth. You can reduce the reproductive index, obviously, by interfering with transmission. And so they're saying that in places where they see this variant, the R value is increasing. However, I would say, and again, this is my theme, you can't use epidemiological data to prove a biological effect of a amino acid change in a virus. You have to do experiments to do that. And that's what they're doing here. They're saying, there is an increase in the transmissibility. It must be because of the variant. Well, obviously, that's a flawed argument. That's not how we do science. The other piece of data is the CT value for the PCR, right? They take nasal swabs from patients, they do PCR, they get a CT value, which is reflective of how many RNA copies are present in the sample. They say there's a decrease of CT of around 2 associated with the new variant. Now, the CT values can go anywhere from 11 to 35 or so. That's the range. 11 being a lot of RNA and 35 very little and probably not infectious. So they're saying in people infected with this variant, we're seeing the CT value decrease. They have more RNA, so that must mean that the virus is reproducing better in them. And they also cite viral load experiments. In other words, they take a nasopharyngeal swab, extract the nucleic acid, sequence it, to convert it to DNA and amplify it, then they sequence it. And then you can calculate the number of RNA copies originally present, and they see they're seeing more RNA copies. So those are mainly the 3 pieces of data that they use to conclude that they have moderate confidence that this variant demonstrates a substantial increase in transmissibility compared to other variants. So there are 2 adjectives in this sentence that I disagree with. Moderate, I don't agree, moderate confidence, and substantial, don't agree at all. I think these are all circumstantial evidences that do not prove anything. You can imagine that R value could change by other things that are affecting transmission. And so you can't conclude it's the actual virus. And as I said, if the virus is introduced into a population by founder effect and it's neutral, it's not going to have any effect on the R value. And the PCR is flawed in itself. You cannot use PCR to measure infectious virus. And that's really what matters. If you want to conclude that this virus is more transmissible, you need to measure infectious virus, and I'll get back to that later. PCR doesn't measure infectious virus, and I can imagine, for example, you have a variant that makes more smaller pieces of RNA in the infected cells, so the PCR shows a lower CT value. Completely flawed argument to indicate that this is leading to increased transmissibility. But they then say we don't have enough data to draw any conclusions about the following. First, the mechanism of increased transmissibility. I would say we don't yet know that there is increased transmissibility caused by these particular amino acid changes. And so they say, for example, increased viral load. Well, you have to measure infectious virus. You can't use PCR to do that. Age distribution, disease severity. I think, if anything, this variant is going to cause less severe disease because of the orphate deletion. And then finally, antigenic escape. So, as I said earlier, the location of these amino acid changes, they're using the wrong terminology as well. The location of the amino acid changes in the receptor binding domain of the spike raises the possibility that this is antigenically distinct, and they've identified 4 reinfections among 915 people with this variant. But tell me how many reinfections you see with the other variants as well. Is it not different? This is one antigenic site, one epitope of the spike. And there are about 20 B and T cell epitopes on the spike. I really don't think that one epitope change is going to make much of a difference. And so they want more data on reinfection, readmission, and case fatality rates, which are apparently being collected, more on the age distribution. And then this third one I think is important to look at, in vitro data on the ability of convalescent sera to neutralize the variant, to see if the amino acid change in the antigenic site impacts the ability of convalescent sera to block infection, which would have, of course, implications for vaccines. But I don't think there are any. As I said, this is one out of many epitopes. It is unlikely that it would make the virus unable to be neutralized, say, by antibodies that are induced by vaccination. And finally, they want to know if lateral flow assays to detect this variant, these would be antigen assays, would be compromised in any way. I think it depends on how you've set up the lateral flow assay, of course. If you're using more than one antibody, you're probably better off. Now, here's what I think about this whole story. I would say I have low confidence that this variant demonstrates a substantial increase in transmissibility. I don't see any evidence for that. I don't see shedding evidence for increased transmission. So if you think a virus is transmitting better, one possibility is that it's more stable in the environment, and that can be tested. Another possibility is that the infected people shed more virus in respiratory secretions. That can be tested. So measure shedding and compare it to people infected with the other variants that are circulating in these areas. Not everyone is infected with this variant. However, NervTag doesn't have this on their list of experiments. And as I said, PCR and sequencing cannot answer this question. Nucleic acid is not infectious virus. This experiment is not going to be easy because you have to have a cohort of people infected with this variant and a variant that's different, predecessor, for example, and compare shedding by isolating virus. And you have to do that in cell culture in a BSL-3 laboratory, of course. And the confounding problem is that when you take a nasopharyngeal swab, for example, you get a slightly different volume from each person. So you have to somehow normalize them when you do your infectivity calculation. Not an easy thing to do. And remember that not too long ago, another variant, D614G, arose and propagated throughout the world. And similar scary stories were told about that one, increased transmissibility. But nothing was ever done in humans to really address that, including the experiments that I'm suggesting. But in the end, I object to the use of epidemiological data to prove the biological properties of a virus. You need to do experiments with the virus to prove that you have increased transmissibility, or at least properties that are consistent with increased transmissibility, and that simply has not been done. In my opinion, all the hype about this variant is unwarranted. Certainly, we have to pay attention to it. We have to study it. But so far, I see no reason why this variant is any different from any others that have arisen. I think we should move on from the scary headlines and get ahead with vaccination programs, which are underway. And that is going to be the way that we get away from this pandemic. I hope you found that explanation useful. I'm Vincent Racaniello, and I'm Earth's virology professor. Thanks for watching.
All right. So, yeah, Vince did a really good job right there. He lost a couple of us, but hey, it is what it is. So I gave you the source that he uses in his display to explain things. You should be able to click directly onto that and get into what needs to be done. Let me see. Am I going to provide you with something else other than that? Nope. That's all I'm gonna give you on that. Everything that you saw here and additional information is in the description. All right. Now, that was a lot to unpack. Not really a lot, but a lot to unpack. Especially if you just new to this information and you haven't been walking with us through this whole entire process. Now earlier you saw, Chavis and myself was having a little bit of a discussion, and there's a couple of things that I want to that I want to put on the screen real quick. I should have been sharing the screen the whole time so y'all could have seen Vince, but I didn't. I didn't want YouTube acting funny again like it did with Sutek earlier. So let's do this. Let's go to live science real quick. And what is RNA? This flexible molecule that you see right here on the screen, we're finna get a whole bunch of pop-ups, tells the cell proteins making factories what DNA wants them to do. Stores genetic information that may help life get it started. All right. So more than just DNA's lesser known cousin, RNA plays a central role in turning genetic information into your body's proteins. This remarkable molecule also carries the genetic instructions for many viruses, and it may have helped life get it started. Together, RNA, short for ribonucleic acid, and DNA, short for deoxyribonucleic acid, make up the nucleic acids. One of the 3 or 4 classes of major micromolecules considered crucial for life. All right. Now, I probably lost a lot of y'all, so I'm gonna let's see if it do it. But here's a great example right here. RNA is a basic way, in a basic way, is the biomolecular that connects DNA and proteins. Let's just do that. RNA connects DNA and proteins. All right. So let's go back. Now, if you're connecting DNA and proteins, right? Why the heck do we need it for our skin? Y'all remember when I showed y'all? No, did I? No, I didn't show y'all this one. But this pretty much from the same site. And there's the illustration, which we'll never get because we ain't there. And then, of course, I can play this, and it's going to say all of this down there. I'm looking in the chat. We heard this mentioned CT values. We could go here. It's a Lancet article, but we just wanted like a general understanding of what CT values are. So there's a document right here. CT values, what they are and how they can be used. You see the date on it, November 2020, so it's the most recent. All right, so there are many factors that impact the use of interpretation of CT values that are generated during real-time PCR testing. Diagnostic laboratories should not include CT values on laboratory reports because it could be out of compliance with laboratory regulations, and they should not be used in any form of patient management. We're going to skip all of the introduction stuff. We're just trying to get an understanding. We're trying to see if we can all get an understanding of what CT values are. So we're going to skip the first 2 parts. I gave y'all the link in the chat to the PDF, and we're going to deal with what it is. What is the CT value? Many NAT tests generate a number as part of the test results for real-time PCR. This is called the CT, or cycle threshold value. Okay, a cycle threshold value is defined as the number of amplification cycles required to reach a fixed background level of fluorescent at which the diagnostic result of the real-time PCR changes from negative, not detectable, to positive, detectable. Now, the total number of cycles required to exceed the established threshold to call a result positive is specific to that test platform and generally ranges from about 15 to 45 cycles. Different tests calculate the CT values differently, and different tests will also count the number of cycles differently. Some tests generate CT values through software installed on the instrument itself. Some require the operator to interpret and define CT value based on the parameters set by the test manufacturer, while others do not generate the CT value that is available or visible to the operator and simply provide a positive or negative test result. In addition, some tests have established the CT cutoff beyond which the test result is considered negative. For others, the cutoff is the last cycle of the test. Now, these parameters are determined by the test manufacturer and cannot be altered by the laboratory performing the test. So now y'all know what CT values are, or a little brief. And the other one is viral overload. Not overload, just a viral load, my bad. So what is viral load? Let's see if we can get something more recent. All right, viral load, just short, simple definition. The amount, well, we're on years age. Let's go back to the top. All right, okay. I should have used this anyway. All right, so viral load and how it is measured. Viral load test measures the quantity of genetic material, commonly RNA, of a virus present in the blood. Several tests are typically done over an extended period, with initial measurements serving as the baseline of subsequent measurements compared to this. So again, viral load test measures the quantity of genetic material, commonly the RNA. All right. So we just want to keep it there on that level. Everything is reverting back to RNA. This is why they made a messenger RNA vaccine, so that it can help tell your body what to do, how to do it, and defend itself when it encounters the S protein, of course, SARS-CoV-2, COVID-19. All right, there we go. So there's more simple. Well, that ain't more simpler. That's still complicated with all the big words. All right, let me stop sharing real quick. We've been online for 47 minutes. Questions, comments, concerns. Questions, comments, concerns. How we looking in the chat? We all got everything in there. There were some keywords that was mentioned. We kind of got them defined, and I agree. I like the way that yeah, I like the okay, the messenger. Okay, let's deal with that. All right, so let's break it down to the last compound. Let's go one more time. Let's go here, genome.gov. All right, notice I keep going to Genome. I keep going to this website that's ran by the NIH. Fauci people. We don't be looking for nothing. All right, messenger RNA again is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. The mRNA is an RNA version of the gene that leaves the cell nucleus and moves to the cytoplasm where proteins are made. During the protein synthesis, an organelle called a ribosome moves along with the mRNA, reads its base sequence, and uses the genetic code to translate each 3 base triplet or codon into the corresponding amino acid. Example here. Traveling on through its journey, right? It's traveling on through its journey. All of this is going on inside of here. You got the cell. If you're looking at your skin, you're looking at a whole bunch of them. Just look at your skin. If you look too closely, you'll start seeing little, weird stuff on your skin. Some of y'all ashy. So you might see them little cracks in your skin. Or you might look at your hand, the top of your hand. You can look at the top of your hand. You got all this stuff going on in your body. RNA, RNA, RNA. So the messenger is only trying to tell you, hey, look, I need you to do something different because there's something out there that you're not used to and you never encountered before. So I need you cells to peep game. I need y'all to get with this program so when you do encounter it, you can create a defense mechanism that will stop it from penetrating you. And we're going to do this all over. So if I come in your right shoulder, I'm going to go throughout your entire body. And I'm going to help you create a defense mechanism against something you never seen before. You can't see it, it's blind. But I know it exists because we're dealing with the gene. We got the genome mapped. We know its strand type. You see where it says the DNA, the strand of the gene? We know what it is. We know we're dealing with these. We're dealing with this. So trust me, I'm going to make sure you good. And I'm going to help you do what needs to be done. Let me fight this battle for you. And then I'm going to see how long I can fight this battle. And if I fight this battle for a long time, I can help you defend yourself against it. But if this thing continues to mutate and a new strand continues to develop from it, and it keeps on attacking, no matter what time of year it is, we're going to have to make some adjustments. And what adjustment we going to make? We're going to make a whole new mRNA vaccine to defend against that strand. If what we have right now does not work. Yeah. That's what I'm saying. So the messenger is like the messenger is like the town whisperer when the king is on his way, or the youngest son in the household when he hear mama pulling up and everybody running rampant. You know what I'm saying? Hey, mama just pulled up. Everybody get to trying to clean up real quick. That's what the mRNA, this is what this vaccine is. The messenger is literally that. It is the messenger. It's a synthetic use of something that's telling the rest of your body, like, hey, you could do this. I'm going to show you how to do this. I'm not going to affect the rest of your body. I'm only just trying to I'm only just trying to help you learn from me long enough, and then I'm going to die out. Because it's going to die out. If you ain't heard, when it get too far into you and all of that other stuff, it die out. Your body does away with it. It takes it's going to take what it want from the vaccine, and then the rest of it don't even exist to it no more. So it don't do nothing else to the rest of your body. It's going to die out. But in that process that happened real fast, between the two shots and all of that, and I say real fast, but it's more like, between the two shots and all of that, it's basically saying, look, I got you, dog, but I need you to go get the booster to make sure that we run through this program legit. Now that we ran through you legit on the second go, hey, We can defend ourselves against anything coming now. We good. But the health officials are still going to tell you that, hey, you still need to mask up and you still need to wash your hands and sanitize, because you could encounter it and shed it and never doing it. We still doing more research on it. You could possibly be you possibly could be infected, right? You could be infected, but you just not experiencing anything from it because your body is just like, psh, you ain't doing nothing. Yeah, we might be infected, but we really ain't got nothing. But that's just basically the truth of the matter when we talking about what this thing going on. So that's pretty much what messenger RNA is. Now, if we need to get a little bit more specific, we can, but that's basically all it is. And I did this the other day with the other video, but the messenger is telling your body like, hey, look, man, you need some help. You can't do this by yourself. Your RNA by itself ain't gonna get it. Your RNA needs a messenger. Your RNA needs a town crier. Let me help you. Because yeah, it's synthetic, but it can do things that we can't naturally do. We can't naturally gain immunity from something we've never encountered before. This is why vaccine, creating new vaccines like this is going to be tremendous for us. So let's go to Mrna real quick, and then we'll wrap up. And let's play this video from Mrna, and let's see how confusing we'll get with Mrna, or if we're able to keep up with what's going on right here. So what I want to do is learn about the messenger's RNA role in the human biology, the instructions it provides direct cells, the body to make proteins, and why we believe mRNA medicines may have the potential to, to do what it needs to do. Boom. Let's go.
My name is Stephen Hogue, and I lead our platform efforts at Mrna, together with our platform chief scientific officer, Melissa Moore. At Mrna, we believe messenger RNA represents an unprecedented opportunity to develop medicines for patients, one that could have a dramatic impact on the course of human health and disease. In order to understand why we believe that, we have to pull back and ask a very fundamental question, which is, what is life and how might messenger RNA impact it? Now, life is really just the flow of information in a biological system. The ebbs and flows of that information dictate health and disease. And that's true in every cell in our body, throughout our bodies, and throughout all forms of life. In order to explore that more deeply, we must follow the flow of information in cells. And in cells, information is stored in the DNA. Now, DNA is a double-stranded helix of tightly wound genetic information that is itself wound around a set of proteins called histones that are themselves then tightly wound around each other into chromosomes. It's analogous to those old tape drives in IBM computers where, in order to access that information, what cells have to do is they have to go find that right tape drive, unwind it, and start to make a working copy. That process of unwinding and making a working copy is called transcription, where a series of proteins will search that DNA, unwind it a bit, bind to the DNA, and start a process of making a pre-messenger RNA molecule. That pre-messenger RNA molecule, when done, will go off and be spliced by a whole different group of proteins called the spliceosome that will edit out certain pieces, leading to a messenger RNA. That messenger RNA is the first fully intact copy, working copy of the information of life that then leaves the nucleus and goes and does all of the work of life. It does that by seeking out ribosomes in the cytosol. These ribosomes translate the messenger RNA into all of the proteins of life, including secreted proteins, transmembrane proteins, mitochondrial proteins, and even proteins that go back into the nucleus and do all the work of storing that information or selecting information to be unwound to make more messenger RNA. So messenger RNA really is the central molecule in all of biology's processes. We call it the software of life. Now, given that power, it's reasonable to ask whether others have pursued it as a therapeutic approach. And in fact, many of the more more exciting technology platforms in biotechnology today are. One example is siRNA. siRNA specifically binds to and degrades messenger RNAs inside of cells. It selects a specific messenger RNA relative to others, thereby changing the flow of information. Another example is gene therapy. In gene therapy, DNA is introduced from the outside and translocates into the nucleus of cells, where either episomal DNA or DNA that is inserted into the genome of the host will go through those same processes of transcription into pre-mRNAs and splicing into final mRNAs that then get exported out of the cell. In many senses, gene therapy is an indirect way to increase or add a specific type of messenger RNA to cells from the outside. So all of these approaches are about modifying the types or amounts of messenger RNA inside of cells so that you can have a phenotypic effect in a disease. Our approach has been more direct. Can we put messenger RNA directly into cells to create proteins, both endogenous proteins and foreign proteins? And if we could, we believe that messenger RNA could be used to make the full set of proteins in life that might allow us to treat an incredibly broad range of human diseases.
Let's go. So let's deal with a little bit more. All right. So mRNA's role in protein synthesis. Step 1 is the double-stranded DNA. Through the process known as transcription, an RNA copy of a DNA sequence for creating a given protein is made. Step 2, parent DNA. This copy of mRNA travels from the nucleus of the cell to the part of the cell known as the cytoplasm, we keep hearing that, which houses ribosomes, we keep hearing that. Ribosomes are complex machinery in the cell that are responsible for making proteins. Then through step 3, then through the another process known as the translation, ribosomes read the mRNA and follow the instructions, creating the protein step by step. Number 4, the proteins. The cell then expresses the protein, and it in turn carries out its designated function in the cell or the body. So using mRNA to develop a new category of medicine, we ain't got to go too far into that. We heard them on a spill on that. And then we're gonna let me drop this on y'all too, because you know I like to drop things on everybody, so everybody's abreast of what we got going on. If you're watching the video on a replay, I'm gonna make sure that this is also in the video, in the description for you to grab, so you can share with family members and friends.
Our platform at Mrna focuses on advancing the frontier of messenger RNA science and technology, trying to take advantage of the intrinsic pharmacologic features of messenger RNA to advance mRNA-based medicines. mRNA is universally utilized to make all of the proteins inside of every cell. So in theory, you can use messenger RNA to make any protein inside of any cell in life. That diversity of applications, the ability to explore all of that biology, is one of the things that we think is most powerful about it.
With biologics, whatever protein is being injected into the patient has to work in the bloodstream. It has to work outside of cells. And only really about a third of human proteins can work in that way. Because we're supplying the instructions to your cells to make the proteins, we can make any type of protein.
That includes proteins that are normally accessible to technologies like small molecules and recombinant protein, extracellular proteins, and secreted proteins or transmembrane proteins. But actually it also includes a whole range of intracellular proteins that are not normally accessible with traditional recombinant technologies.
Another advantage is that when your own cells are making the proteins, there are a lot of what are called post-translational modifications. There are decorations that are put on the protein by your own cells. And because your cells know the right decorations to put on, they're going to just do that automatically. Why would we want to do that? Well, because in the cell, a lot of functional proteins act as complexes. They act as multiple protein complexes, a little community of proteins that, in order to get their job done, have to work together. And so when we want to either replace some activity where it's a multi-protein complex, that's the problem, or we want to make a vaccine where the thing that your immune system needs to recognize is a multi-protein complex, we can just supply all of the mRNAs that encode all of the proteins in that complex. And your cells then just can make those proteins and construct it and present it.
Proteins are really optimized for a given state. And so being able to make proteins in the right tissue, in the right place in the cell, allows you to make proteins that are more stable and that can last longer and have a more potent pharmacologic effect. All messenger RNAs are comprised of 4 simple nucleotides in different combinations to write different codons or words that then are used to make all of the proteins of life. And those 4 bases ultimately are combined in just different ways. That ability to make an incredibly diverse number of molecules with the same set of ingredients following a relatively standard approach confers incredible flexibility and speed on our platform.
One of the things that you want in a drug, obviously, is for it to be reliable. And what we know, because of its very nature, a single mRNA sequence encodes a single protein. So if we give an mRNA, we know exactly what protein it's going to encode, and it's always going to make that protein. Another feature is dependability in terms of dose. And you want
She said something that was very important. No matter what they code, it's going to make that protein. No matter what they code, it will make that protein. This is the cutting edge. This is the cutting edge technology that will help advance vaccine medicines. Well, I'm just gonna say vaccines in the future. So when a new pandemic comes, as long as they can understand how this is responding, then they can deal with it from an RNA perspective. If it's something outside of that, then some tweaking and some unconventional things that have to take place. But right now, they own it.
want a proportional response from your drug. So if you give one dose, you get a certain response. If you increase that dose, your response goes up in a predictable manner. And that's exactly what we see with mRNA. The protein output is proportional to the amount of mRNA that we give. Now, one of the things that's also a really nice feature is that we don't have to give very much mRNA.
Messenger RNA is amplified. An average messenger RNA will be translated thousands of times. So one mRNA can result in hundreds to thousands of proteins.
And then finally, mRNAs are confined to the cytoplasm. They do not get into the nucleus, and so they do not interact with the genomic DNA, and there is no worry about causing any changes to the genome.
Mm-hmm. Kill the pseudos. Say that one more time, lady. Kill the pseudo.
Worry about causing any changes to genomic DNA, and they interact with the nucleus plasm. They do not get into the mRNAs are confined to the cytoplasm. They do not get into the nucleus, and so they do not interact with the genomic DNA, and there is no worry about causing any changes. Are confined to the
thousands of proteins.
And then finally, mRNAs are confined to the cytoplasm. They do not get into the nucleus, and so they do not interact with the genomic DNA, and there is no worry about causing any changes to the genome.
Trying to tell y'all, team. Trying to tell y'all, man. Trying to tell y'all. Straight up and down. Trying to tell y'all this for a while. No matter what, I've been trying to tell y'all that. You know what I'm saying? So we had people that were skeptical about the vaccine and this, that, and the third. Well, you know, that was in 2017. You know what I'm saying? That was in 2017. Let's go look at Pfizer. So Moderna been on their game. What about Pfizer? What about AstraZeneca? Will we be getting news from AstraZeneca? Will I have news tomorrow from AstraZeneca's release? Will we have a release on AstraZeneca? We would like it, because if we get it, we in the door.
New York, and now
I ain't doing that. I ain't doing that. NBC, baby, I ain't gonna get me right there. Stop right there. So look, ladies and gentlemen, we are emerging. We're on the cutting edge of doing all the things that we need to do to make sure that our people are safe. I appreciate everybody tuning into Science with Shawn this evening. You know what I'm saying? I'm gonna look in the chat and see if we got any questions. If I ain't got no questions, then I'm gonna slide out. We got 24 people watching. Can we get some more likes? We've been at it for an hour and 10 minutes, and we learned a lot today. We unpacked a lot in a short amount of time. And I don't want to extend the video out because I want people to be able to digest it. And it's some rewindable moments in here that will help advance knowledge. We trying to be scientific. We are trying to advance knowledge. Now, I could have shared screen early in the video, but I didn't. We shared screen at the end. We got what we needed to do out of Science with Shawn. We got our lesson. We got our lesson twice. What up? What up, Dave? Let's see if we got a question in the chat. YouTube rejected your comment. This usually means they think it's spam. Try changing it a bit. YouTube is heavy on also team. I think from now on, I won't be dropping links in the chat because of that right there. YouTube's been like blocking people from commenting in the chat. They calling it spam. And I don't know how you on your own videos and you educating your people and you trying to give them the sources. So for me, I'm just going
educating your people.
My bad. I'm just going to I'm gonna continue to put all my stuff in the description. And you know, after the show is over or whatnot, or during the show, if you're following along with the show, you can go ahead and you can grab all of them for yourself, because they're gonna be in my descriptions every time. Also, in the description, we have our FamilyConnectApparel.com, the link to all of our merchandise, Amazon.com to get your copy of Killmonger's Corner, Part 2, and of Killmonger's Corner, Volume 2 of the journal. You also can, make sure you go to KofiPiesideTV.com to learn the market and purchase all 7 of our books. That's right, I said 7 books. We have them. Chilling. I got my letter for COVID. Okay. What up? Yeah, Dave, speak on that. Okay. Dave got his letter. That's what's up, Dave. Dave got his letter. But forget all that. Did you get your books? Did you get your books, Dave? Tell us about that. I just want to say pseudos made the Mayans wrong a second time. The world did not end. Yep, we still living. We ain't get no superpowers. What superpower did you get? Let me put that in the chat. Shawn superpower is reading. So yeah, man. The books will be at my house tomorrow. Cool. Yeah, I knew it was coming up. I know it was coming up, fam. So that's what's up. And did nobody else get no superpowers? Yeah, YouTube say you spamming yourself. You need some vision. I know it's a little delay, so I'm trying to wait, see what everybody's saying. Man, I got a homeboy named Tavon, man. He's from New York. He's pseudo as heck. If I showed y'all what he be posting, man, he be tripping. He be posting some weird stuff. And I don't understand how he's so pseudo. And it's always YouTube videos. Everything he posts got a YouTube video. He needs to stay off. He needs to stay off certain YouTube channels. I ain't gonna tell him to stay off YouTube, but he needs to stay off certain YouTube channels because he getting all the wrong information. And that's what YouTube should be censoring and blocking and all that other stuff. They should be focused on that. I got soul energy. I can detect stupid people. Yo, y'all crazy. Everybody else get no superpower? Come on now. Y'all mean to tell me y'all ain't get no superpower? My feelings is hurt. I really thought I was a super, I don't want to say it on here. I thought I was going to be with it, man. Thought I was going to be the one. I thought I was going to be able to have one of those soul glow moments. I got BS vision. I see right through BS. Absolutely. I know you was over there watching your boy, man. He still alive? Your boy is still alive with her? He's still lying over there, I should say. Let me check and see if I can get y'all some. Oh, I did want to share something with y'all. I did. Hold on. We ain't done. Nah, I'm going to do it tomorrow. Yeah, I'm going to do it tomorrow. We're going to get into that. You know what I mean? It's going to hold y'all for about a half an hour. Science with Shawn. Yeah, I ain't going to drag that out. I'm going to do that tomorrow. That's a fact. Let me keep this bite size. Yep. All right, yo, Shawn, how is your best friend? Oh, he's cool. He's laughing and stuff. Let me put that on the chat so the viewers can see you asking me that question. Yeah, he's cool. You know, it's the same stuff. He hasn't really did too much. He's just watching what he doing and how he doing, and you know, just trying to maintain and get back to some normalcy in his life. Yeah, so he's been able to take care of whatever he need to be taken care of. He said, I got an uncontrollable laugh when a pseudo is serious. Boy, you crazy. For the people that will actually watch this in the archives, I really appreciate you tuning in and hanging out no matter the length of the actual video. If you feel like this information is useful and resourceful for you, not only yourself, but members of your family and friends and co-workers or whoever it may be, somebody in your social media network, would you please share this information with them? Spread the word. These are things that we actually can do for ourselves when disseminating information and trying to make sure that our people got what they need in order to survive. You know, let's do that. let's do that and let's make it happen. Share it, like it, share it, tell a friend, tell a friend, tell a friend, keep telling friends, keep trying to get the word out, man. Look, this is ain't about me. I ain't doing it for you know, to get you to tune in and then I don't give you what you're looking for. You know, I want you to tune in so I can give you exactly what you need. This is episode 12. This is episode 12. So, you know, let me check the chat one more time and see what my people got going on. Look like we done commenting. Tomorrow I'll have a quicker lesson tomorrow. We won't be too long. Pop up in the middle of the day, possibly. I don't know. I haven't came to that actual conclusion. But I hopefully this video is informative and you guys learned something from it today. Because, you know, knowing is half the battle. You know, we got to continue to know and do everything that we need to do to make sure that we cool where we at. You know what I'm saying? That's how I go. Ladies and gentlemen, the books is in. The books are in. Yep. They in. They're here.